Hey everyone, welcome back. Last time we ended talking about nutrient availability and the different soil properties and how that affects nutrient availability and the trade-offs that are there between the availability of water and nutrients and the propensity for leaching or cation exchange based on those soil properties. I want to talk today more about nutrient uptake itself and how these nutrients are actually entering the plant. And so when we talked about the primary growth and roots, we talked about the zone of maturation. And so that zone of maturation is the area where the nutrients are actually taken up, where water and nutrients are taken up. And those are all taken up through those root hairs. And so here's a nice diagram showing a germinating seedling. We see that zone of maturation with all of the root hairs. And I just want to remind you that the root hairs are essential for increasing the surface area for nutrient and water uptake. Remember, the roots are really harvesting these really diffuse resources, those nutrients in the soil and the water in the soil, and bringing them, concentrating them in the plant. So that increase in surface area is really an important part of this. And the root hairs themselves, those outgrowths of these epidermal cells in that zone of maturation, really increase the surface area a lot. The other thing is that root hairs also have a very large number of transmembrane proteins. Why is this important? Well, this is important because remember that plasma membranes are semi-permeable. And so some things can pass through plasma membranes, water being one of those things that can easily pass through the plasma membrane. However, most of the nutrients that are there actually in the soil actually require transmembrane proteins, channel proteins, antiporters, symporters, etc. Those transmembrane proteins to actually move them from the soil water and move them into the inside of the cell, into the symplast of the cell. Cell walls, as we know, are porous, and water and the nutrients dissolved in the water can enter into those porous cell walls and travel through the apoplast all the way down to the Kasparian Strip at the endodermis. And so I'll talk a bit about that. So water with those nutrients can get into the root cortex and travel through the apoplast and through those porous cell walls, but only until it gets to the Kasparian Strip. And at that point, it does need to enter the symplast of the cell and cross the membrane. And so those transmembrane proteins are important there as well. But root hairs, many of those nutrients entering the cortex go into the symplast through transmembrane proteins, the large number of transmembrane proteins that are found in along the root hairs themselves. So lots of transmembrane proteins to move these in. And so that facilitates the passage of those nutrients into the plant. And so I just want to again remind you that we have a situation where outside of the root hair, outside of that cell in the soil, we have a very low concentration of our nutrients. Here's a potassium ion shown here, a nitrate ion shown here. These are at very low concentrations outside of the cell, and the plant is concentrating these inside of those cells. And so that the concentration gradient is actually more concentrated inside of the cell, less outside of the cell. And so we actually, for most of these these nutrients that are dissolved in the water, they need to come in against their concentration gradient. And in order for that to happen, the plant has to establish that electrochemical gradient. So we've talked about this already with with phloem loading and unloading. and the importance of the proton pump to be able to move solutes against their concentration gradient. Well, that becomes very important here as well. So those root hairs have many proton pumps associated with them as well. And that is to establish this membrane potential. The membrane potential is a combination of electrical potential and that electrical potential is actually established by having different number of these positively charged protons outside of the cell than inside of the cell. And so we can see in this example here, we've got a lot of positive ions outside of the cell, more negative ions inside of the cell. There's actually an electrical potential across this gradient. And then of course there's a chemical the gradient that's there as well. And that is the fact that we actually have a concentration gradient, more protons inside or outside here than inside. And so it's a membrane potential is a combination of the electrical potential established by the protons and that concentration gradient. And so as such, we call this an electrochemical gradient because it's both electrical potential as well as that concentration gradient. And that's established by the proton pump. And importantly, the proton pump uses ATP. And so this costs energy for the cell to be able to establish this electrochemical gradient between the outside of those root hairs and the inside of the root hair. And so this electrochemical gradient actually affects the way that different ions, cations or anions, are taken up by those root hairs. And so if we look at cation uptake, we've got the example of this potassium ion here. So there's our cation in that potassium ion, and this needs to flow against its concentration gradient. And so in order for that to happen, the cations are taken up through channel proteins. So they enter the cell through channel proteins. Using, or I should say following, that electrochemical gradient. So the proton pump is established, and the proton pump establishes that electrochemical gradient from the outside to the inside, and then these positively charged potassium ions can then just pass through these channel protons following that electrical gradient. And so they're moving along the electrochemical gradient from positive to negative. And so they can, in that sense, they can actually move against their concentration gradient. So we see that they're more concentrated inside the cell than outside of the cell, but we're just moving through a channel proton. And so this is sort of, so while the potassium is passively following the electrochemical gradient, this costs energy for the proton pump to establish that electrical gradient that these cations will follow. So cations can enter through channel proteins when there's an established electrochemical gradient. How about anions? Anion uptake is a little bit different because the electrical gradient actually is going from positive to negative, as we showed before. And so anions use symporters to move these in. So remember that symporters take advantage of the electrical gradient, the higher concentration of protons outside of the cell than inside of the cell. Those protons then flow back through the channel or through the cotransporter, through the symporter, and they move them, they move the nitrates or the negative ions, those anions, with them. So in this example, it's a nitrate. So they use the symporter to flow those negative ions, so anions, against their concentration gradient. So it's not only against the concentration gradient, it's also against the electrical gradient, so against the electrochemical gradient, to move those in against that gradient, both electrical and concentration gradient. So, and they're flowing with, so cotransporter, symporter, they're flowing with those hydrogen ions as they flow back into the cell. And so with that, with using both cotransporters like these symporters, as well as the channel proteins, like we saw in the example of the positive ions manipulating the electrical gradient, the proton pump can establish electrochemical gradients that allow for both cations and anions to enter the cell through specific transmembrane proteins. So this is all great, but one of the things that is important to recognize is that these nutrients, these ions, the cations and anions, the nitrates, the phosphates, the potassium, calcium, etc., all those essential nutrients are really diffuse in the soil. And so while the root hairs increase the surface area, it actually, there's still oftentimes you know, a lot of searching that goes on to find these diffuse resources. And there's a really special association with fungi in most plants that allow them basically to extend their root system and harvest these very diffuse resources. And this association is called mycorrhizal. Mycorrhizal fungi are fungi that have a symbiotic, and I can go farther and say mutualistic relationship with plant roots. And that symbiotic relationship is that the fungi, the fungal hyphae, so these thread-like hyphae that are going throughout the soil system, extend the plant root system immensely. And the fungi themselves uptake very efficiently both nitrogen and potassium from the soil. And so they are, and these are two essential nutrients that are necessary for normal plant growth and development. But they're also extremely diffuse in the, sorry, did I say, I said potassium, I meant phosphates. And so they're also extremely diffuse in the environment, in the soil. And so having this, this basically exponentially increased root system by associating with these fungal hyphae as well, this is a really efficient way. And so while the mycorrhizae are providing nutrients to the plants, the mutualistic part of this is that the plants are actually providing sugars. They're providing photosynthate, starches, sugars to the fungi themselves. And so there's a win-win situation for a win-win situation for both the fungi and for the plants themselves. And most plants have a mycorrhizal association. The mycorrhizal associations come in two flavors. Those are called, and I'll show you the differences between these. These are called ectomycorrhizal and arbuscular mycorrhizal fungi. Arbuscular mycorrhizal fungi, you'll sometimes hear these referred to as endomycorrhizal fungi. Arbuscular is the more correct term in these. But these are two different ways, and different groups of fungi, of course, do this a little bit differently, but two different ways that fungi are associated in this mutualistic relationship with plant roots. And so in ectomycorrhizal fungi, we see ectomycorrhizal fungi are fungi where the hyphae penetrate between the cells. So hyphae penetrate the intercellular spaces between cells. And so this is through the apoplast. And so we know we've got this very porous cell wall that are these porous cell walls, and we see the fungi, these ectomycorrhizal fungi, actually come in through the apoplast and are in the cell, those spaces in between the cell wall and in that porous cell wall. And these all grow through them. They're getting sugars from those plant cells, from the plant cells that are there, and then they're also from the starches that are stored in those cortical cells, and then they're providing those phosphates and nitrogen to the plant in the form of these dissolved ions in water. Oops, I'm not sure why that's duplicated. Sorry about that. And then our second type is actually arbuscular mycorrhizal fungi. And arbuscular mycorrhizal fungi, also called endomycorrhizal fungi, actually penetrate the cortical cells. And they grow into them. And the reason it's called arbuscular refers to tree-like branching. And so when they penetrate, we see them growing in through those cellular spaces and then penetrating those cortical cells. We see these branching branching forms of the fungal hyphae inside, and these are these arbuscules. We see them growing into those cortical cells, and this is just a more direct interaction, and so a bit more efficient in terms of obtaining those sugars and delivering those nutrients and water to the plant. But still a mutualistic relationship within fungi, with fungi and plants. So this mycorrhizal relationship is very important. And that's basically, you can think of this as extending the root system of these plants. We'll see that in a little bit that this mycorrhizal association can actually get exploited by other plants, and we'll show you that in examples of some parasitic plants. Okay, the other piece of nutrient uptake or modification to nutrient uptake that I want to talk about is an important one, and these are mechanisms of ion exclusion. And this is especially important in things like heavy metals or salts, things that are, as we've talked about, deadly, lethal to, toxic to the plant themselves. And so there are ions in the soils that you don't want to let in. So heavy metals being some of them. Although some of these, like copper and nickel, are definitely essential nutrients in high quantities. They're toxic to the plant, and so most of these are positive ions in the soil, like zinc, copper, nickel, etc. These metal ions. And when this actually this diagrams on the right actually show some phenotypes associated with copper toxicity. And so while I'll just put in here, while some of these are essential in small quantities, so micronutrients, in large quantities, they're toxic. and create really bad results. And so because of that, there are both passive and active mechanisms for excluding these. Now, I know we've talked about this already with respect to the Kasparian strip, and I'll remind you of that here. But so plants have active, meaning they cost energy, and passive mechanisms for exclusion. Okay, so let's take a look at some of those mechanisms for excluding the excluding these ions, and I'll just mention first the passive one that we've mentioned before, and this is the Kasparian strip. And so, as we know, water with all of its dissolved nutrients, ions, can move through the apoplast and make its way to the endodermis. But at the endodermis, we have this whole suberized strip of cells that we call the Kasparian strip. This blocks then anything from entering through the entering the xylem, entering the vascular cylinder in the root. Through the apoplast, and so things have to then cross that membrane to get into the symplast of the plant. And this is where the Kasparian strip can passively exclude mechanisms. And the way that it does that is that the Kasparian strip lacks channel proteins associated with certain ions that we want to exclude. And so here, this shows an example of a sodium ion or a cadmium ion while there are channel proteins, as we've talked about, that allow things like nitrogen through or calcium through, essential nutrients that we want to get in there. We want to limit the amount of sodium, and we want to limit the toxic heavy metals. And so these don't have channel proteins and are then excluded from entering the xylem and being spread throughout the plant. So that's one way where they'll never enter the symplast, through this passive exclusion by having a lack of channel proteins associated with these ions. But there are also active mechanisms that exclude, and I'll talk about two of them here. I'll just separate those. The first of those is one for actively excluding the many metal ions, and these are called metallothioneins. And what metallothioneins are is these are actually proteins that are made by the plant cells that actively bind and sequester toxic heavy metals. So these are special proteins that are made by the cell, and so this costs energy to make these special proteins, and their whole sole purpose is to go in, bind onto these free ions that maybe got into the cell, like extra sodium ions or copper ions or other toxic heavy metals, and sequester them. And so it binds them and inactivates them. It makes them not available to the rest of the cell. And so in that way, they are not going to be toxic to the plant. The other way is that the ions are sequestered in the vacuole. So remember that plant cells have a vacuole, and that vacuole has a membrane around it. That membrane is called the tonoplast. So this is the membrane. And this membrane actually has a lot of channel proteins, specifically antiporters for specific ions. And so this is really common for salts. And so the example here is shown for sodium, where we have outside of the vacuole in the cytosol, we have a low concentration of sodium. Inside, we're actually moving the sodium ions into the through the tonoplast into the vacuole, inside the vacuole, to concentrate them. And so that then takes it out of the cytoplasm and sequesters these in the vacuole, making them not toxic to the plant. And so the ions are moved into the vacuole to sequester them. And they're moved in against their concentration gradient using a proton pump, establishing an electrochemical gradient to move these against the concentration gradient into the tonoplast. This one's been studied quite a bit. And like with other plant systems, the way that this is actually recognized as an important mechanism of ion exclusion is through experiments done with Arabidopsis. Arabidopsis is a model plant system because it has a very quick generation time, and there are many known mutants in this. And so here we have this is a slide showing plants that have additional antiporters present. And so they've actually done an experiment to transform these plants so that they make more of these sodium antiporters along their tonoplast. So these have a lot of them. And we see that with increasing concentrations of salt from A to E, we don't see very much difference in the growth habit of the plant at all. So these are actually able to take that salt that will be toxic in those high quantities and sequester them all into the vacuole. The control plants have no additional antiporters, and what we see with the increased concentration of sodium, we actually don't see much of an increase, we see a decrease in the growth of those plants. So we can see that as that salt concentration increased, the plants are doing poorer, and that's because they're not able to. Sorry, there's our salt concentration. We're not able to actually handle that much salt and sequester that into the tonoplast or into the vacuole without having more of those antiporter proteins made. Okay, so those are both a passive and then a couple of active mechanisms for dealing with for excluding certain ions. We'll just leave that at that. Another really important piece to nutrient uptake is actually how bacteria play a role in taking up nutrients. And so nitrogen, although atmospheric nitrogen, and two, it's the most abundant, nitrogen's the most abundant element in our atmosphere, it's not available to plants in its atmospheric form. And two, plants cannot use it. And so plants actually need nitrogen converted into nitrates or ammonia or ammonium to be able to take those up and use that nitrogen. Nitrogen is an essential macronutrient, and so they need it in large quantities, but although it's very available in the atmosphere, we can't actually get it to the plant without converting it. Bacteria play a huge role in fixing what we call fixing nitrogen. And so taking that atmospheric nitrogen and making it into a form, nitrates or other nitrogenous organic compounds like ammonia and ammonium that the plant can actually use. And so there are nitrogen fixing bacteria in the soils that convert atmospheric nitrogen into these usable forms. And they create this nitrogen cycle that's really an important part of nutrient uptake for plants. Some plant groups have taken this one step further and actually have a mutualistic relationship with nitrogen fixing bacteria. This is most common in the legume family. This is the family that includes all of our beans, but it's also found in other families, and I'll show you a distribution of this on the phylogeny. But this is really common in legumes, and I'll just say among others. I will mention that the legume family is one of our largest families of flowering plants with around 20,000 species. And so it's possible that the success is lent a bit to having this symbiotic relationship with nitrogen fixing bacteria. I'll also mention that that legumes, because of this, are often the first colonizers of nutrient poor habitats. And so after disturbances that maybe strip the nutrients from particular soils, legumes are often those first colonizers, because they can take nitrogen from the atmosphere through their nitrogen fixing bacteria. And so these, in legumes specifically, they have a nitrogen, a symbiotic relationship with a group of bacteria called Rhizobium, and these are nitrogen fixing bacteria. And the mutualistic relationship is that the plant gets nitrogen in a usable form, so usable nitrogen, and the bacterium gets sugars and a home. And so throughout this process, what happens is that the plants that are infected with this rhizobium, or these nitrogen fixing bacteria, create these root nodules. So along the roots where root hairs would have been, root hairs are formed into these nodules. And if we look inside of these nodules that are shown here, we can look inside and see that they are filled with bacterial colonies. And so we've got the rhizobium bacteria inside of these nodules. And this is part of a process this is sort of the end result of the process for these plants being infected by these symbiotic bacteria. I'm not going to hold you to learning this whole, to memorizing this sequence of events, but what I want to show you here is that it's the root hairs themselves that are manipulated. And so the plants are releasing a signal. This is a biochemical signal, flavonoids, into the soil, and that actually attracts the rhizobia, the nitrogen fixing bacteria, to the plant root hairs. Once those start to associate with the root hairs, this root hair actually curls up and it creates what's called the bacteria create an infection thread. And that infection thread penetrates the cortical cells and helps form this nodule that is eventually the home of these nitrogen fixing bacteria. I mentioned that nitrogen fixing bacteria are, very common in the legume family, but they're also found in several other families. Alders are a tree that you're probably familiar with that have a different group of bacteria. Frankia, I think, are the name of those bacteria that are associated with alder roots. But there are several different bacteria and several different groups of plants that have a mutualistic relationship with nitrogen fixing bacteria and create these root nodules like we see here. And there is a cool paper from a few years ago, this is 2014, that showed that these different groups of plants that have relationship with different types of nitrogen fixing bacteria actually came from a single evolutionary innovation that's driving this nitrogen fixation in angiosperms. And this paper utilized one of that, I think I showed you a big phylogeny that we did looking at freezing tolerance in plants. And so they actually used that same phylogeny of around 30,000. They trim it down to from that same phylogeny. And they use that phylogeny to investigate the distribution of nitrogen fixation. And what they found is, while the legume family is the large majority of this, that's this whole big white bar here, it's all the legume family, and they have all of their nitrogen fixers with rhizobium as the bacteria. There are several other groups of nitrogen fixing bacteria, several other families here's betula, the birches, and that's where alder falls in that family. But there are these several other groups, and some of them have nitrogen fixation and some of them don't. And that's what's made this confusing as whether these are independent origins of that or not, is that there seems to be a lot of gains and losses. What these guys found using some comparative methods comparing the rate of the gains and losses of these traits is that what is the most likely scenario is that there is a precursor trait. They don't know what that trait is. That evolved once. So the best model predicts there's a precursor trait that evolved once, and then that made it basically these are pre-adapted to evolve nitrogen fixation in this whole big group of that we call the nitrogen fixing clade, this whole big group of angiosperms. So while they have different bacteria that they're associated with these different origins, or these different nitrogen fixing lineages, they likely evolved from one innovation that happened a long time ago in the common ancestor of this whole nitrogen fixing clade. Okay. I'm gonna end this talking about, so that's our nutrient uptake, some special situations with exclusion of ions and mycorrhizal associations and other mutualisms with bacteria for nitrogen fixation. I want to end this with a few nutritional adaptations and just talk briefly about special situations where special situations that are different in plants that are in other groups. I'm going to pause the video here and I'll come back just to give a little break in this. I'll pause the video here and I'll come back and we'll finish off the nutritional adaptations in the next video. Thanks.